Hi there, this video outlines basic enzyme structure and its function. So an enzyme is protein-based, and it's usually relatively large to other molecules in the cell, and it has a pretty distinctive three-dimensional shape because there's going to be sort of a lock and key mechanism. So here's our enzyme, and remember that enzymes are proteins. Typically, a couple of exceptions because this is biology, but for the most part, enzymes are proteins. And on the enzyme, there is an active site where the substrate can bind. So where I drew that, I placed it right here. And so for a substrate to bind, and a substrate might be, for example, just a sugar that your body is breaking down or your cells are breaking down. So when I say the fancy term of substrate, I'm just referring to what's entering the reaction. So I'll put an example of sucrose. That's table sugar. So you eat something that's maybe very sugar intensive. And this enzyme is going to catalyze its change or reaction. In this case, for sucrose, I'm going to talk about a breakdown reaction, but you could also have a different set of reactions where you're building molecules as well. So this enzyme will catalyze the breakdown, in this case, of sucrose, for my example, into the reactants. And so sucrose can be broken down into two simpler sugars, which are fructose, which many of you are familiar with as fruit sugar, and glucose, which again many of you are familiar with because it's blood sugar. So those would just be examples. Okay, so the substrate will bind to the active site. So if I draw my sucrose molecule here, and it could be any substrate, it needs to be able to fit into that site. So typically there's an area on the substrate itself that's called the binding site, and this is kind of where that lock and key comes in. So the substrate will bind to the protein-based enzyme. So I'm drawing an arrow here to the binding site. And then the reaction will occur. And so in this case, you might be breaking it down because sucrose is a disaccharide, it's a larger molecule. You could be breaking it down into two smaller molecules. So we could be breaking it down into our glucose and our fructose. So these reactions will occur without enzymes, so you can have a breakdown of sucrose. You can just leave it in a jar on your table. Eventually, over time, it'll break down, but you probably know that the breakdown rate would be so incredibly slow that it would be meaningless for the body, because if you think of how long it would take to break down, just sitting on your table or maybe sitting in a glass of water where it would start to dissolve, it's a long time, and your body needs to get at it a lot faster than that. So enzymes work by catalyzing the reactions. So a reaction like this would be what's called catabolic, or a breakdown. And if you remember from other readings, that's typically going to be exergonic because we're releasing energy, and we're getting at the energy that's inside of that sugar, that disaccharide sucrose. You also can have anabolic reactions as well. So a building reaction. So let's say you're building up a protein from different amino acids in your diet. So those are typically going to be anabolic, but those are also enzyme catalyzed as well. And those are typically also going to be energy requiring or energy consuming, and those are endergonic. It takes energy to build protein. So the way enzymes work is that, again, I mentioned that the reaction would happen what we call spontaneously on its own, but it would be so slow. So it takes some energy to get reactions started, regardless of whether they're catabolic or anabolic. And so that beginning piece to get that started is called the activation energy. And that actually gives the cell a little bit of control, because you don't want to just spontaneously break down, let's say, your muscles, unless you're really desperate for calories and your body's trying to get at energy that's stored in there. So you want to have some control. So by having this activation energy, it takes a little bit to get it started, and I equate that with getting on a roller coaster and you go up, 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 uphill. It takes a long time, and then when you get to the top, then the rest of it's pretty fast. You go through the roller coaster pretty quickly, but you have to build up that energy, that potential energy to go through the roller coaster. So what enzymes do is they lower the level of needed activation energy. They make it so that there's less energy needed in order for this substrate to be broken down. So it's actually going to lower it down. So this would be no enzyme. And this is just kind of a sketch, not necessarily to scale of the change, depends on the enzyme. So it would lower it so that you'd only have to get up here for the reaction to occur. So this would be activation energy with enzyme. And going back to that roller coaster example, that's the slow part of the ride. That's when you're going up, up, up. It takes a long time. So imagine if you only had to go a third as high up that hill to get enough energy for the roller coaster to proceed, how much quicker the ride would be, because you would that slow part would be over. Or maybe if it's a roller coaster with multiple uphill build up, you know, by lowering that hill, it's going to happen a lot faster because you're going to get to the height that you need to much quicker. So that actually changes the height of energy that's needed. A couple other things about enzymes: they can be denatured. And what is denaturing? Well, they can be permanently broken down or permanently changed by extreme conditions. And you know, we've experienced this on a less severe level, most of us, with fever. So fever, when the fever goes up, the temperature goes up in our body. That's what fever is. So an increase in temperature. And that makes us feel pretty lousy. And what that's actually doing is starting to denature our enzymes. So why do we do that? That doesn't sound like such a good maneuver. The idea is that if you have a virus or bacterial infection, that you're also denaturing their enzymes, and that they're going to be a little less resilient. They're a smaller organism, maybe dependent on a smaller selection of enzymes, and so that you make that temperature so high that their enzymes get denatured. However, if your temperature gets too high, then you do have that problem where you are going to have some sort of, you know, whether it's you go into shock or coma, you can get very, very ill if that fever gets too high. So temperature is one way. pH is another common one. So acids can do that. So we can go to cooking too. If you cook an egg, okay, that egg is never going to go back to the way it was. You've changed the shapes of the proteins with heat. pH can do that as well. So acids can really change that change permanently, and even other things like salts. And usually the focus is on temperature, pH, but mechanical agitation. And going back to my egg example again, let's say you make a meringue or something where you need to whip egg whites. They fold into a brand new shape. You kind of whip them and whip them. They increase in volume. You can never get them back to that original egg white that when you first cracked it out of the shell. So that's denaturing. So again, we use that in a positive way with fever, and we use that in foods, right? We cook foods to help break things down. We preserve foods with acids. So if you look at your spaghetti sauce, that's typically got a lower pH. Salsa, sometimes foods have added vinegars. Think about if things are pickled. All of those do things to lower the pH, and then therefore inhibiting microbial growth. So the lasting time will be a lot longer. Okay, so that's all I have there. Again, just review that enzymes are protein-based. They have what's called a binding site, and it needs to be that exact shape. And going back to denaturing, when that folding changes because the temperature's gone up or the pH has gone down, that is going to permanently change the shape of this enzyme such that it will no longer work properly. And different enzymes have different ranges of temperatures and pHs they can exist at. So if you're a microbe that lives in a hot spring, your enzymes function at a much higher temperature than, let's say, a human who's at that very narrow kind of moderate body temperature range. So protein binds to the substrate. That substrate then is converted to reactants, and again, it can be breakdown. That's one example. I gave an example there. Could be building molecules as well. You're building new cell membrane. You're making new DNA in your cells. Those would be anabolic and what we call endergonic. And enzymes work by lowering the energy of activation. That's where the slow part is of the reaction. Once you get there, it's quick, 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 quick. So the idea is if you lower the energy of activation, you can keep these reactions happening at a fast enough rate. One other thing I do want to mention is that enzymes do not actually enter the reaction, so they're recycled after each use. They do wear out eventually, but they are recycled, so they can keep, for as many molecules of sucrose as you have, it'll keep going and going and going. And that enables the body to keep the enzymes at pretty low concentrations so that you can have a variety of enzymes readily available. I thank you very much, and please contact me with any questions that you may have.